Inside every living cell, machines too small for microscopes cut, copy, repair, signal, and defend. They are proteins. For decades, biology could read the instructions for making them, but could not see the machines themselves. Then, an artificial intelligence system began predicting their shapes at a scale no laboratory could match. Not perfectly, not magically, but fast enough to change biology's map. AlphaFold helped crack one of biology's defining puzzles and opened a new era in biotechnology. A protein begins as a line, a sequence of amino acids written like molecular code. Inside the cell, that line becomes a shape, and shape is everything. A pocket may capture a drug. A surface may bind a virus. A mutation may destabilize the machine. For most of modern biology, knowing the sequence was not enough. Researchers had the letters. The hard part was seeing the object. A gene might reveal a disease mutation or a pathogen's weakness. But, without structure, it could feel like repairing an engine without opening the hood. For decades, the most reliable path was experimental. X-ray crystallography, nuclear magnetic resonance, and cryo-electron microscopy revealed structures in extraordinary detail, but each required the right sample, expensive instruments, and deep expertise. These methods remain essential. They are among science's most powerful tools, but they can be slow, difficult, and selective. Some proteins are flexible, some are embedded in membranes, some exist only briefly, some are overlooked. By the early 21st century, biology was producing sequences at industrial scale. Genomes could be read faster and cheaper than ever before, while the structures encoded by those genomes remained far harder to obtain. The archive of life was expanding. The molecular map was still incomplete. In 1972, Christian Anfinsen received the Nobel Prize for work showing that a protein's amino acid sequence contains the information needed to determine its folded structure. In principle, the code contains the shape. In practice, predicting the shape was one of biology's hardest computational problems. A protein can twist through an astronomical number of possible configurations. For decades, computational prediction improved, but slowly. Then came CASP, the critical assessment of protein structure. Prediction. Every 2 years, teams entered a blind challenge, predicting structures that had already been solved experimentally, but not yet publicly released. No shortcuts. No looking up the answer. For years, progress was real, but incremental. Then, in 2018, DeepMind entered with AlphaFold and made a major leap. In 2020, AlphaFold 2 changed the field. At CASP 14, AlphaFold 2 achieved a median GDT score of 92.4, with many predictions approaching experimental accuracy for single protein domains. Around the same period, ROSE-TAFold, led by David Baker's lab, confirmed that learning-based structure prediction had entered a new phase. AlphaFold was not an isolated miracle. It was structural biology meeting deep learning, evolutionary data, and decades of public experimental work. The reason AlphaFold worked was not that it simulated every atom folding through time. AlphaFold does not watch a protein physically fold from start to finish. It learns patterns. It begins with the amino acid sequence, then searches for related sequences across the tree of life, building a multiple sequence alignment. This is evolutionary memory. Across millions of years, if two amino acids sit near each other in the folded protein, mutations in one position may be compensated by mutations in another. AlphaFold reads those correlated changes as clues. Distant points in a one-dimensional sequence may be neighbors in three-dimensional space. The history of life contains hidden geometry. An attention-based neural network reasons across sequence, evolutionary patterns, and residue pairs, refining a structure until a likely fold emerges. And just as important, AlphaFold reports its confidence. Some regions appear sharp and stable, others are uncertain, flexible, or unreliable. The output is not experimental truth. It is a high-resolution hypothesis. Return to the researcher at the screen. The blank space is no longer blank. A predicted structure appears, not a final answer. A starting point. For drug discovery, AlphaFold can help identify possible binding pockets, prioritize targets, and guide experiments, especially where experimental structures are limited or absent. For human disease research, predicted structures can help interpret mutations, A change buried in a protein core may destabilize the fold. One near a pocket may affect binding. One at an interface may disrupt a complex. For rare diseases, where every clue matters, structure can turn an abstract genetic variant into a physical hypothesis. During the COVID-19 pandemic, DeepMind released predicted structures for several SARS-CoV-2 proteins. These models did not replace virology or structural biology, but gave researchers early hypotheses to test. In sustainability, structure prediction is helping scientists study enzymes for cleaner chemistry, carbon capture, and plastic degradation. AlphaFold accelerates the question. The laboratory still tests the answer. Proteins are not statues. They move, open, close, bind, and release. A single predicted structure may miss the motion that makes a protein function, and structure does not automatically reveal function. A protein's role can depend on timing, location, chemical modifications, partners, environment, and regulation. AlphaFold is strongest for many single-chain globular proteins, but biology is often messier. Protein complexes can be difficult, especially when interactions are transient or weak. AlphaFold, Multimer, improved predictions for complexes, but uncertainty remains. Intrinsically disordered regions may not fold into one stable shape at all, yet they can be biologically essential. Proteins with few evolutionary relatives can be harder to predict because the model has less evolutionary memory to read. Earlier, AlphaFold systems did not fully model small molecules, post-translational modifications, membranes, or the crowded chemistry of the cell. AlphaFold 3 expands the scope to include interactions with DNA, RNA, small molecules, ions, and other biomolecular components. That is a major step. But it is also a reminder. As predictions become more ambitious, validation becomes more important, not less. The danger is not that AlphaFold is useless. The danger is that a beautiful visualization can feel more certain than the evidence behind it. AlphaFold changed access. With the AlphaFold protein structure database, predicted structures for more than 200 million proteins became available worldwide. For labs without expensive structural biology infrastructure, this was a new scientific atlas. Not a replacement for experiment, a navigational layer, but access creates new questions. If a predicted structure helps lead to a commercial therapy, who owns the downstream value? The AI provider that made the map? The public database? The institution? The company? The team that validated the target and made the drug? Public prediction and private invention now exist in the same pipeline. There is also bias. AI models learn from existing biological data, which reflects decades of unequal research investment. If the world is not careful, AI may accelerate biology where data already exists and leave neglected areas behind. Then there is biosecurity. Structure prediction alone does not create a pathogen, but powerful biological design tools can become part of a broader dual-use toolkit. The same knowledge that supports medicines and diagnostics can also raise concerns around harmful biological agents. This is not science fiction, it is governance. Return once more to the researcher at the screen. The structure rotates, a mutation glows, a pocket appears, a hypothesis forms, but the decision does not belong to the model. Human control still matters. What experiment should be run? What uncertainty is acceptable? What result is enough to publish? What target is safe enough? What knowledge should be open, restricted, patented, or regulated? This is the responsibility network. The researcher interprets, the engineer builds, the database distributes, the company develops. Regulators set boundaries. The scientific community validates or rejects. Imagine a future where an AI proposes a protein design, a robotic lab tests it overnight, a model updates the design by morning, and a company begins development within days. If something goes wrong, where is the decision? In the model? The lab? Dataset? Approval process? Market incentive? Human authorization? Distributed intelligence can accelerate discovery, but distributed discovery also distributes accountability. AlphaFold did not solve biology. It changed the surface of the unknown. The life code is not cracked in one moment. It is opened layer by prediction, experiment, caution, and imagination. If you enjoy deep dives into the technologies shaping our future, consider subscribing to HiTechnomy. This is HiTechnomy. Look beneath the surface of the future. Join us next time as we explore another technology, system, or breakthrough shaping our world. Powered by Alligence Labs.